Hello, and a very warm welcome to our Cell and Gene Therapy Insights Live 30 webinar. Accelerate your cell and gene therapy workflows with next-generation analytical tools. Joining us today is Dr. Chris Hager, who's the Director of Application Science for the Analytical Solutions Division of Bio-Techne. Following his presentation today, we'll have a Q&A session. You can ask a question at any time throughout the event by using the Ask a Question box, and we'll try to get to it during the session. If you'd like more information on the topics discussed today, please take a look at the Resources tab for the useful ebook. And with that, I'll hand over to Chris for today's presentation.
Thank you, Elisa, for the introduction, and I would like to thank our attendees for joining me today to learn about how to accelerate your cell and gene therapy workflows with next-generation analytical tools from Bio-Techne. Bio-Techne has a broad portfolio of manufacturing and analytical solutions for biopharma and cell and gene therapy applications. These include a wide variety of GMP-grade reagents, including cytokines, growth factors, and small molecules, and media and supplements. We also have next-generation reagents for cell activation as well as non-viral gene editing under GMP conditions. We also offer a wide variety of critical research reagents, including industry-leading immunoassays, antibodies, proteins, and ISH technologies. And lastly, through our Protein Simple brand, automated analytical instrumentation, which will be the focus of today's talk. I'm here to talk about how we can accelerate your cell and gene therapy workflows with next-generation analytical tools. Let's start by taking a look at a generic cell and gene therapy workflow to orient ourselves. Starting with a viral vector and gene of interest, a targeted viral vector is composed. This assembled viral vector requires characterization for various qualities, as we will discuss today. For gene therapy, where viruses like AAVs are used for in vivo delivery, the next step is gene therapy transfer assessment and then patient delivery. For cell therapy, cells are modified with lentivirus carrying the gene of interest and then expanded. Contaminant screening is a necessary part of the process prior to introduction of the final product into patients. In both cell and gene therapy, after delivery, a response is expected which can be profiled. There are 4 analytical platforms to discuss in the context of these workflows: Simple Plex and Simple Western, Maurice, and MFI. Let's get started with Simple Plex. Simple Plex is an automated ELISA, and our instrument shown here, Ella, is well suited to replace any ELISA in your cell and gene therapy analytics arsenal. Ella runs ELISA assays in about an hour and a half and only requires 25 microliters of sample volume per well. The innovative cartridges shown on the bottom right come in a variety of formats to provide sample, input, and analyte flexibility, and are supported by a broad assay menu. These cartridges come with built-in standard curves for the analytes, saving precious assay space for samples and also imparting a higher level of standardization than is typically associated with ELISAs. The platform boasts high sensitivity and wide dynamic range, as we'll see today, among some of its top qualities, with excellent lot-to-lot consistency. And lastly, transferring Simple Plex assays to other sites is a breeze, meaning you can easily transfer the assay as part of a work packet to another lab or site. Let's take a look at the Simple Plex cell and gene therapy assay menu. Let's start by aligning Simple Plex with this workflow. First, let's look at Simple Plex in the context of potency. The USP 1047 for gene therapy products states that when the biological function of the expressed transgene exhibits a broad range of activities or only generates semi-quantitative results, the ELISA or other immunological or biochemical readouts can be used as a surrogate potency assay. We have 2 assays currently on the menu to address this part of the workflow, one for AAV2 and one for P24 to detect lentivirus. Specificity and robustness to matrix effects are major advantages of ELISA, but in considering the long turnaround time, moderate throughput, As well as the labor-intensive sample preparation, it's challenging to implement for routine quantification of capsid titer in a process development setting without automated liquid handling. We've partnered with ProGen to put their antibodies in our Simpleplex assay cartridges to make an AAV2 assay. You can run up to 72 samples per cartridge, and the standard curve is built in, so you can add the extra samples that you need. Shown below is the standard curve with a detection range between 3 times 10 to the 6th capsids per mil and 1.3 times 10 to the 10th capsids per mil, with a dynamic range about 3.6 logs. The assay has great correlation with the ProGen ELISA, providing confidence in the product's ability to measure viral titer accurately. And the assay performs quite well in precision assays, shown here against the ProGen lyophilized control in blue and the ATC reference material shown in orange. The assay can also accurately measure AAVs in complex samples as demonstrated by the excellent spike in recovery observed, and this can be done over a good range with a dilutional linearity from 1 to 2 to 1 to 32 in several commonly encountered sample matrices, including clarified lysate, column eluates, as well as bulk product. Similarly, we have a fantastic Simpleplex assay for measuring P24, which is an HIV protein found within lentivirus. Quantification of P24 can be used to estimate lentivirus titer. Shown here is the built-in calibration curve with a limited detection of 0.67 picograms per mil. The assay has strong performance with inter-assay CVs under 10% and intra-assay CVs under 7.1%, and shows good linearity and recovery in several sample types. Now, the assay performance shown here doesn't clearly spell out lentivirus work, but we do know that the kit is being used for this. In fact, shown here is a pre-press article from GSK where they're using this very assay for lentivirus titer according to the manufacturer's instructions. Characterization of cultured immune cells following activation, gene transfer, and expansion is complex and leverages both cell surface markers and soluble secreted biomarkers. Simplplex's multi-analyte cartridges run on the Ella instrument provide an ideal platform for fast and accurate quantification of secreted markers in cell culture supernatants. Customizable assay patterns eliminate the need to run multiple ELISAs, and assays with up to 4 logs of dynamic range allow markers of varying abundance to be measured from a common sample dilution. These panels include our T-cell NK cell activation panel, our cell cytokine secretion panel, as well as our chemokine secretion panel. Here's a great example of how Bio-Techne can come together to provide a more complete solution for you. To look at T-cell activation with Simpleplex, we set up an experiment with the expertise of our Genome Engineering Services group, who used our TC Buster transposase non-viral gene editing technology to introduce a CD19 CAR into T-cells. These T-cells were then used in a cell-killing luciferase-based assay against 2 cell lines, K562 myelogenous leukemia cells, which don't express CD19, and NALM6 acute lymphoblastic leukemia cells that do express CD19. On the left are the results of the T-cell-mediated cancer cell lysis assay, where the cancer cells that express CD19 are lysed. On the right, we screen the conditioned media of T-cells incubated with either K562s or NALM6 cells on Simpleplex. T-cells that can recognize the CD19 positive cells become activated and release granzyme B, TNF-alpha, interferon gamma, and IL-2. Granzyme B is released at quite high levels near 800 picogram per mil, whereas IL-2 increases only to approximately 40 picogram per mil. Coming back to our workflow and adding the Simpleplex cell expansion and characterization panels, we can now move on to contaminant screening for cell therapy products. Specifically, today we're going to talk about our new HEK293 HCP assay. We have partnered with Cygnus to once again bring an industry-leading host cell protein assay to Simpleplex. For those cell and gene therapy folks coming from the biopharma space, You might know our CHO-HCP assay that features the Cygnus 3G CHO-HCP antibody. Well, we've done it again, this time to help get your HEK levels under control. Introducing the Simpleplex HEK293 HCP 3G assay. This assay features 3-log dynamic range with an LLOQ of 1.64 nanogram per mil. The assay has excellent correlation to the Cygnus ELISA shown on the bottom left. Let's now take a look at how the assay performs for monitoring process steps for HCP removal. Looking at 3 different processes, the Simpleplex assay closely mirrors the Cygnus ELISA data, giving further confidence for those already using the Cygnus reagents but looking to move to a faster platform. We've gone on to show that the assay exhibits excellent spike in recovery across a wide variety of process samples. Importantly, the assay performs well with different sample matrices and has excellent dilutional linearity across the 5 most encountered samples. This is a new assay, so we're really excited to get into the hands of more researchers to get their feedback and to support their impurity testing. I'd like to now move on to our Simple Western platform. Simple Western is the next generation CE Western platform to support your cell and gene therapies. Simple Western is an automated capillary-based Western platform that requires as little as 3 microliters of sample per analysis. It's highly flexible and can be thought of as an open platform with multiple detection modes, including chemiluminescence, 2-color fluorescence, as well as an ability to measure total protein. The platform is highly sensitive, generating assays with a wide dynamic range. In addition, Simple Western features replex on some instruments to double the amount of data per run. And lastly, just like Simpleplex, due to the automated nature of the platform, assays can be easily transferred to other sites. Looking again at this workflow, it's easy to peg Simple Western to a number of steps here. And that's because Simple Western is an open platform, providing complete flexibility for cell and gene therapy developers and manufacturers. Those potential uses include critical attribute monitoring, where Simple Western can support a number of assays for viral vector characterization. For cell characterization, as needed, and while I don't have a specific example today, really any protein target where there's an antibody should work. For contaminants, Simple Western can be used to look at host cell proteins as well as other impurities. In addition, getting down further after the cell or gene therapy has been administered, Simple Western can be used for response profiling. And frankly, even farther upstream before the vector is even made in R&D. Today, I would like to provide a few examples of how Simple Western can support the CQAs for viral vectors. Simple Western can be thought of as a gene therapy multi-attribute machine and provides 6 assays on one platform. These assays are purity, identity, and capsid protein ratio, empty full status, physical viral titer, and protein expression potency. Of even further benefit is the fact that many of these attributes can be measured in a single run. Let's briefly look at 2 examples. For the first example, I'm going to show you here a quantitative AAV assay that provides identity, purity, and capsid protein ratio at the same time, basically combining 3 of the assays from the previous slide. For this assay, we are leveraging replex. In the first probe of the capillaries, we will look at the identity of 4 AAV samples, and in the second probe, purity. So shown here are 4 AAVs, AAV1, 4, 7, and 9. In the first probe with an anti-VP123 antibody. From these data, we can see that AAV1 and AAV4 aren't recognized by the antibody we're using, but AAV7 and AAV9 are. Additionally, AAV9 and AAV7 appear to have different sized VP3 proteins. From these data, we can estimate the capsid content, or viral protein ratio, which is 8 to 2 to 1 and 11 to 1 to 1 for AAV7 and AAV9 respectively. And then in replex, we look at the same capillaries for total protein, which shows that these samples have a decent amount of non-AAV protein in them, shaded here in gray. Again, these data can be quantified automatically by our software and used for normalization or for purity estimation. I'm really excited to share this next example with you. For these data, we are leveraging a replex assay on a Jess instrument. Replex allows for a strip and reprobe of the capillary for double the data per sample in the same run. In this run, we first denature and separate AAVs and then interrogate them with an anti-DNA antibody. In this example, it's shaded in green. The apparent molecular weight of the DNA is quite large, as expected for an AAV plasmid, and agrees with some other DNA work we've done. The capillaries are then stripped and reprobed with an anti-VP123 antibody from ProGen to measure the viral protein amounts as well as the capsid ratio in each sample. So again, 2 assays in one. In this case, for this series of AAV9s, we obtained an average capsid ratio of 13 to 1 to 1 for VP3 to 2 to 1. The data shown can be quantified and used to estimate the amount of DNA in other samples. I've additionally added a lane view representation on the far right to help better illustrate the data sequence. In probe 1, we have the chemiluminescence detection of the DNA, and in probe 2, we have the NIR detection of the VP123 proteins. Lastly, we've tested this assay with several other serotypes and observed the same general trend when comparing empty and full AAVs. Maurice is the next generation capillary electrophoresis platform to support cell and gene therapies. Maurice provides the gold standard IMAGE-CIEF for charge heterogeneity analysis of proteins with absorbance and native fluorescence detection. In addition, Maurice has CESDS for direct detection and purity analysis of proteins, which is quite useful for AAV capsid ratio as well as purity. You need your data fast, and Maurice can provide it with data ready in as little as 15 minutes per sample, which allows for faster decisions as well as rapid method development. We've designed the instrument to be easy to use and have cartridges that dictate the mode run. Our software is 21 CFR Part 11 compliant with optional and power control, and methods on Maurice are easily transferred to other sites, just like our Simple Western and Simple Plex technologies. Maurice is an ideal late-stage viral vector characterization tool. And is best suited to support viral vector characterization and critical quality attribute monitoring with purity, identity, capsid protein ratio, empty full, as well as stability assays. Starting with Maurice CESDS, which is a direct SDS page replacement no-brainer. AAV product purity is a critical quality attribute to monitor. Here we are looking at 4 preclinical AAVs from 2 masked serotypes named M and S from an industry collaborator. There are a number of peaks observed in addition to the core capsid proteins VP3, 2, and 1. Those peaks, down to a 0.1% impurity, can be quantified and used to assess overall product purity. For these 4 samples, purity was greater than 91.9%, and the average capsid ratio was 6 to 1 to 1, which wasn't unexpected when discussing the data with our industry collaborator. In addition to the purity assessment and capsid protein ratio, a novel peak was identified in sample S2, which we named peak 5, at a 0.8% total peak area level. These data show a powerful example of how Maurice can support identity in addition to purity and viral protein ratio, all in the same run. IMAGE-CIEF is a powerful identity tool. We have developed 2 methods to look at AAVs. In the first, our capsid protein characterization method, we denature the AAVs prior to analysis. This method is platform and can be used for identity as well as stability indication. Shown on the left is an example of a panel of AAV serotypes using the capsid protein characterization method. These samples, again, were denatured and diluted to 4 × 10⁻11 GC per mil prior to analysis. For all 8 serotypes, a minimum of 3 peaks can be seen, and in several samples, more than 5 peaks are readily quantifiable. Each serotype not only has its own series of peaks, but its own set of PI values as well as peak ratios, showing that the method can be used as an identity assay. These data are quite interesting given the similarity between the serotypes. While most of the AAVs exhibit a similar profile, there are certainly some interesting serotypes to call out. First, AAV5 is clearly the most acidic in this method, and AAV6 is the most basic. If we look at this table published in 2021 in the journal Viruses, AAV1 and AAV6 are 99% identical. If we look at just these two serotypes, we can see the difference by IEF. AAV1 and AAV6 differ by only 6 amino acids, shown here. I calculated the PI of each 6-mer to show how much more basic the AAV6 peptide is, which explains our data and confirms how powerful isoelectric focusing can be for AAV identity. To compare the two image CIEF methods, I have selected AAV8 and 9. These serotypes are 93% identical by the chart on the previous slide, and in the capsid protein method, they align quite nicely. But there's an important difference, and AAV8 has 2 more basic species than AAV9. Let's look at the same sample now when they aren't denatured prior to analysis in our intact method. In this case, the overall PI of each AAV has shifted to the right by about 1 pH unit, and AAV8 and AAV9 no longer align, but have different overall isoelectric points. So the intact particle method is also an identity assay, but the intact assay also has a unique feature to it. As I mentioned earlier, Maurice Image CIF features dual wavelength detection. And I'm going to show you how we can expand the intact method's utility past an identity assay. Maurice provides absorbance detection at 280 nanometers, which, if we look at the absorbance spectra for DNA, shown in red, and protein, shown in dark blue, we see that the contribution at 280 nanometer is more strongly influenced by DNA. Maurice also has a native fluorescence filter, which is centered closer to 350 nanometers, where the signal is exclusively from protein. So if we compare the absorbance and native fluorescence signals together, we should be able to deconvolute the DNA and protein signals coming from an AAV in our intact method. Shown on the right is an example of a pair of commercially available AAV9 samples, an empty and a full from Virovect, where we analyze them with our intact method. Shown on the top in blue is the absorbance of the AAV9 full sample. In black is the native fluorescence, which is looking, again, only at the protein signature and is overlaid. There's very good overlap between the two. However, when we look at the nearly empty AAV9 sample on the bottom, we observe significantly less absorbance, in this case shaded in orange, but the sample still has a substantial native fluorescence signal. These native fluorescence signals can be quantified as well as the absorbance signals, but the native fluorescence signals can be used to normalize any differences in loading between the samples. We're really excited about this potential and look forward to seeing more of it used in your hands. Moving to our last analytical instrument of the day, MFI is the next generation particle imaging platform to support cell and gene therapies. MFI can support the USP and FDA mandated particle counting for biopharmaceuticals, namely the USP 787 and 788, and is used commonly for counting biopharma product aggregates. The MFI features direct dynamic sample imaging of particles between 1 and 300 microns with powerful CFR 21 Part 11 compliant analytical software. Particle data is available in as little as 6 minutes per sample, and like our other platforms discussed today, our MFI platform and method can be easily transferred to other sites. MFI can support cell and gene therapy by monitoring particles in both cell and gene therapy workflows. In addition to being an ideal subvisible particle aggregation tool, MFI has been shown to be stability indicating as well as useful for monitoring impurities in cell therapy workflows. Let's quickly take a look at an example. For this MFI example, we are going to bring in another Bio-Techne product, our clouds. Clouds are dissolvable microparticles that can be used for cell activation and expansion. Today we're going to be looking at the human NK cell expansion kit. These dissolvable clouds feature CD2 NK-P46 microspheres and readily dissolve in the presence of our 1x release buffer. Shown on the bottom left is an example of that measured on our MFI. The big blue bar in the center are the clouds particles themselves, and then all the way on the right is the clouds particles in release buffer, where there are essentially no particles measured on the MFI. Now, coming out of an NK expansion, when it's time to dissolve the clouds, we can look and compare before and after the addition of release buffer. And because MFI can easily distinguish the two particle populations in the same sample, as shown in the inset images on the right, the amount of residual clouds, if any, can be monitored as part of the expansion process. In this example, shown in the middle are the NK cells in release buffer, and then on the right is the NK cells in the absence of release buffer. So a dramatic loss of the NK particles upon addition of release buffer, giving you essentially a pure cell population at the end. In summary, Bio-Techne provides innovative solutions to meet your cell and gene therapy research and manufacturing needs. We provide premium analytical instrumentation through our Protein Simple brand that can analyze multiple attributes for your cell and gene therapy products. Simpleplex supports viral particle titer, residual host cell impurity analysis, and measures cell activation and cell expansion. Simple Western supports 6 assays on one platform: identity, purity, capsid ratio, empty full, viral titer, and protein expression potency, but has many other uses in the cell and gene therapy workflow as well. Our Maurice CE platform is a dual-mode instrument ideal for formulation and late-stage characterization, with CESDS and image CIEF for capsid ratio and purity, as well as identity, empty full, and potency, respectively. And lastly, our MFI particle counter is an ideal imaged subvisible aggregate tool for both cell and gene therapies. With that, I would like to thank you for your attention and recommend you visit our website at biotechne.com for more information. And with that, I'd like to hand the mic back over to Elisa for Q&A.
Great presentation. Thank you, Chris. So as I said, we're now going to move on to the Q&A. We've got lots of questions coming in. First question I've got here. How does your empty full assay work with novel developed capsids outside of the parental panel that you have?
Yeah, hi. That's a good question. Yeah, so I think this is referring to our Simple Western data that I showed, and where I showed a number of serotypes. And we have at this point, it is a new assay. The application note actually just published yesterday. But we have thus far looked at one pharmaceutical AAV that's a non-wild type variant, if you will, and that also works well with the empty full method. So we're encouraged that at least in that first try with a non-wild type variant that we had that success, and we look forward to others testing it out with their program material soon.
Great, thank you. And are Simpleplex assay panels fixed, or can users design their own panels?
Yeah, that's another good question. So the Simpleplex assay panel or assay menu can be customized really to what you need. And I showed some examples with the cell therapy panel where you can choose individual cytokines, and we will build cartridges with the analytes that you need all on the same cartridge.
Brilliant. Is there a list of antibodies suitable for Simple Western applications like the ones you showed in the presentation? what resources are available to guide assay development on that Simple Western platform?
Yeah, we have an antibody database on our website that has, I think, nearly 3,000 antibodies that have been verified to work on Simple Western. With respect to the antibodies I described today, the AAV viral protein antibodies from ProGen are listed on our website as well with the working dilutions, and the anti-DNA antibody that I described here is in the application note and will be also available on the website.
Great.
And sorry, just to add, and it is a product from Bio-Techne, the anti-DNA antibody.
Perfect. And can you tell us a little bit more about Simple Western's replex technology?
Sure, and I realized I didn't answer the second part of the last question, which is about assay development, and I'll hit that and then come back to the replex real quick. So assay development, there's a lot of great resources available through our Learning Academy, which is really geared towards helping customers teach themselves. And we also have a fantastic field application science team to help with your assay development as needed. And then moving on to the replex. Replex is really an elegant strip and reprobe, if you're familiar with that concept from traditional Western blotting. But in this case, it's part of our automated capillary Western blot workflow. And so it's just adding another row of reagents to our plate, and then the instrument will automatically strip the capillaries of the first immunoprobed primary and secondary and allow for a second probing of that same capillary, giving you double the data.
Great, thank you. And I think we've got time for one more quick question. Which instrument is preferred for full empty analysis?
Yeah, that's a good question. So we have two examples I showed today, the isoelectric focusing method on Maurice, and then the size-based separation on our JEST Simple Western platform. I think they both have solid strengths to them. The Maurice method is a direct detection, a little bit more amenable to downstream, but it is isoelectric focusing, so you may have to develop a method a little bit more specific for your AAV than what I showed for AAV9. The size assay seems a bit more universal in that respect, although it does rely on immunodetection. So if your DNA packaged material doesn't react with the antibody, for example, or the viral protein antibody doesn't detect one of your VPs, those could be considerations in why you would choose one or the other. But both powerful tools. Absolutely.
Well, brilliant. Thank you, Chris, for answering those questions. Thanks to the audience for some excellent questions. I think that's all we've got time for today. So any questions that we didn't get to, we will reply by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. I think all that's left then is to thank Chris once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.